So within our tertiary structure, we've got all those different properties that contribute to it, and I still will say that the image your textbook has does a really good job of showing all of those. The primary one that we're concerned about when we're looking at protein folding in both tertiary structure and quaternary structure is the hydrophobic interaction. Okay? So that's going to be the one you're primarily responsible for. That's the one you should really watch out for. So types of questions you could see for hydrophobic interactions within tertiary structure, what do you think? How might I put a question together on the test?
Where would you find a hydrophobic interaction?
So we could do something as directly as that. I would probably push a little bit more application-based and say which of the following amino acids would you find in a hydrophobic interaction or on the inside of a protein. Right? So yeah, you could go through and list off a whole bunch of them. Remember, I only told you had to memorize 5, so I'm going to have to put a little bit more work in on my end to make sure that I don't just list off the 3-letter codes for each of them or even the names, because that would then assume you had them all memorized. So what you'd probably end up seeing is structures. You'd see, and I would probably even further abbreviate it and just say these are the R groups. Which of these R groups would you most likely find on the interior of a protein? Okay. So yeah, your hydrophobic are the ones you're primarily responsible for with tertiary structure. So we've got an example that your textbook went into. They talked about myoglobin and hemoglobin. A little bit better. So we got the structure of the myoglobin there in green, the polypeptide, and in it looks like red, we've got what's known as a heme. Okay? That heme is really the active site for myoglobin, okay, and for hemoglobin for that matter. That's really what's controlling its function. So what is the function of myoglobin or hemoglobin? Maybe you know that one better. Hold oxygen. Okay? So the idea is that if we take a look at that heme, so your heme is the organic molecule on the outside of this. Okay? So we're looking at this whole exterior being your heme. And then in the middle of that, you've got the iron atom. Okay? That iron atom, being positively charged, can coordinate negatively charged things like oxygen, sort of negatively charged. Okay? Oxygen, if we looked at its Lewis structure, has a whole bunch of lone pair electrons, so it can coordinate to the metal center. Okay? And then that metal center can then float around through the bloodstream and transport that oxygen from one position to another. So then that kind of begs the next question. If oxygen already binds to the iron in heme, why do we have a protein at all if the heme actually works to hold the oxygen? A couple answers we could come up with on this.
Wouldn't you need it to be hydrophilic and transfer?
The heme needs to be water soluble so that it can float through the bloodstream. If we take a look at the structure of that heme, is that water soluble? Probably not. We've got an awful lot of covalent bonds in that structure. It's probably going to be very hydrophobic, not going to sit well in water. Okay, so that's a very good reason, okay, that you guys should be able to pick up already. So that's good that you came up with it first. What's the second reason?
Does the protein help carry more oxygen?
Nope. With myoglobin, it's not a bad idea. With myoglobin, it only has, I think it's just that one heme, and the heme is the only thing carrying the oxygen. What do we want this protein or the heme to do?
Easily release it.
It needs to be reversible. It needs to be able to get rid of that oxygen atom. If it can't get rid of the oxygen atom, then great, it binds the oxygen, and then what?
Yeah.
Okay, if it never gives it up, then it defeats the purpose of transporting it, because now you're just transporting this giant massive block around that happens to have oxygen attached to it. So we have to be able to get the oxygen off. The protein helps to allow for the oxygen to bind less tightly to the iron atom. If it binds less tightly, that means it's a little bit easier to reverse, and we can pop the oxygen on and off. Okay? I don't know the exact details of your myoglobin, but it might even change its conformation to help push the oxygen atom further off. Okay? I know for sure that, oh shoot, I just forgot what the amino acid was. I want to say it's something like histidine. It's very, very close to that iron atom core, and that prevents the oxygen from binding end on. So our oxygen atom would actually hit that iron atom directly out of the plane at us. In that perfectly perpendicular situation, it's now bound very, very tightly. So histidine comes in and gets really close to the iron atom, preventing the oxygen from binding end on, and it binds slightly crooked. That slightly crooked situation allows for it to bind less tightly. We can pull the oxygen on and off a little bit easier. Okay? So kind of a neat molecule for that. Questions about the myoglobin? Okay. Oh, I guess one of the other interesting facts about myoglobin was up at the top. It was the first protein to be solved using Xray crystallography. It's kind of neat. With Xray crystallography, I'm not sure I emphasized that enough on Tuesday. It is a rather tedious process. You have to get a single perfect crystal of the protein. Okay? If you don't have a perfect crystal that doesn't diffract light appropriately or the X-rays appropriately, and you get a scattered diffraction pattern as opposed to a very neat one. Okay? And neat is, of course, a relative term. So X-ray crystallographers are known for patience in the process of generating these crystals. So to get a single crystal of a protein, you can look on the order of a successful one, probably in days, to get a single crystal appropriately enough. And that's with a known protocol. Without a known protocol, you're potentially looking at a PhD dissertation on getting that single crystal and that X-ray crystallography graph interpreted, okay, as a PhD thesis. Okay, so it can take a very, very long time. Why might it take a long time? How do we get perfect crystals?
Very slowly.
Think about salt. A perfect crystal is sodium chloride, sodium chloride, sodium chloride all the way through. Very, very regular lattice structure. Are proteins particularly regular? No. So to get them to fold together or come together in an appropriate fashion to allow for them to have a single perfect crystal tends to be very, very difficult. So they tend to tweak the buffer situations, acidities, different other compounds to try and help that crystal structure formation. So it's a tedious process, but the results are kind of neat. It still comes to the same idea. You have to be able to get that in a position. I don't know enough about the electron microscopes, but I'm pretty sure they still have to be stationary proteins. If that protein is moving at all, you're going to blur any of your images. Yeah, and we get just the outside, whereas the X-ray crystallography goes all the way through the whole thing. So you can get what the interior of the protein looks like, full three-dimensional image. Quaternary structure is the next step up. Now we're taking several polypeptides, fully folded tertiary structures, interacting with each other to generate your quaternary structure. Okay? So it's really a significant step up from our tertiary structure, whereas our tertiary was kind of a weird variation of our secondary. Quaternary is very, very clearly different. You're looking at multiple polypeptides. So the example I always like to go back to, because it's the one that just struck me as most interesting, was the ATP synthase. You're looking at, see if I can remember these right, 1, 2, 3, 4, 5, 6, I think 7 different proteins coming together in varying amounts. For instance, within this top portion, there's 2 primary proteins, but there's multiples of 3 in them. Okay. So you're looking at a pretty massive complex of proteins coming together to generate that structure. The next one down below represents hemoglobin. We're looking at the structure of hemoglobin. How did you know that? Did I actually say it there? Yeah, that would have been nice. Okay, so in hemoglobin, it's a more complex version of our myoglobin. Our myoglobin was just our tertiary structure with a single heme. If we take a look at hemoglobin, if we look really carefully within there, you'll notice that we end up with 4 hemes. Okay, so what we're dealing with in that case is 4 separate polypeptide chains that have now arranged around each other to generate the overall quaternary structure of our hemoglobin. Okay. Yes?
Is it basically just 4 myoglobins?
I don't think it's the same as myoglobin. I think it's a different protein, but I don't know that one for sure. Okay. Hemoglobin has some more interesting properties. Number one, in comparison to myoglobin, it can carry more oxygen, okay, because it has 4 hemes. So there's 4 times the amount of places where oxygen can bind. Okay. Another interesting side effect of this is that we can get what's known as an allosteric effect. Okay. One peptide chain can affect another peptide chain within that quaternary structure. Okay. In the case of hemoglobin, what is that effect leading to?
Not the ability to carry oxygen.
Kind of the ability to carry oxygen.
Once you get one, it's easier to get the next one.
Yeah, it builds. As soon as we put one oxygen into the hemoglobin, it changes its 3-dimensional structure, which changes the 3-dimensional structure of the other peptides around it. That allows for oxygen to bind more easily in one of the other peptide situations, to one of the other hemes. It then further changes its structure to allow another oxygen to bind more easily. Okay, so as we increase the concentration of oxygen, we increase its ability to hold oxygen. So we're looking at an allosteric effect. That structural change is increasing that ability. Okay? Kind of makes sense? So if we go through and take a look at graphs, because graphs are always so fun to deal with, what we've got here is the percent saturation and the oxygen pressure. And so what we're evaluating is those 2 curves and what they mean. So what does it mean in the case of myoglobin? Yeah, myoglobin's going to grab oxygen very, very, very quickly. Okay, so it has a very tight bonding pattern with our oxygen.
There's
Okay.
still 4 subunits for myoglobin.
Sorry?
Is there still 4 subunits?
No, myoglobin was 1 unit.
Oh, okay.
Yeah, hemoglobin was the 2. What happens in the case of hemoglobin?
You have that quaternary structure.
Okay, we have a quaternary structure, but why does it not just jump straight up as a straight line? If we take a look at our myoglobin, you notice that it goes from point 0, it goes virtually straight up. Okay, fits a straight line. If we looked at the hemoglobin, why does it not go straight up?
Because it needs more oxygen.
Nope.
It can hold on to more than just oxygen.
That's not going to explain this, because this is looking at the percent saturation of oxygen. So it's it's the allosteric effect. Once we put a single oxygen on, okay, or if we have no oxygen present, it's not going to bond oxygen very tightly. So we get a relatively flat line to begin with. But as we bind more oxygen, what happens? It starts to increase the affinity for oxygen, and we start to see a sharper line come off of that. Okay, eventually, as with both of these, what happens? They start to level off because we've now fully saturated the protein. It can't hold any more oxygen. Okay. Hemoglobin has a lot more dependence on both oxygen as well as a bunch of other factors. Acid concentration, CO2, bisphospho, let's just say BPG, I already forgot it, and chloride ion. Why do we have all this extra dependence for hemoglobin?
Because it doesn't only carry oxygen, it carries other gases.
It can carry other gases, but there's another important reason behind it.
Is it just the elevation too? Like,
Where is hemoglobin? Where is myoglobin? Hemoglobin is typically found in the blood transporting everything. Where is myoglobin typically found?
the tissues.
It's in the muscles and in the tissues. What's the role of myoglobin? Storage of oxygen. The role of hemoglobin, transportation of oxygen. So with myoglobin, we want to hold that oxygen until we absolutely need it. With hemoglobin, we want it coming on and off pretty much as quickly as possible. We're trying to transport it from one position to the other and then get rid of it as quickly as possible. Okay? So in our hemoglobin, we're going to be highly dependent on the concentration of CO2. Why?
That's what we're breathing
That's what breathing.
That's what we're exchanging. Okay? So if we go to the muscle system, we're going to be releasing that CO2 into the bloodstream, and our hemoglobin has to pick it up and then be able to transport it back to the lungs. What happens at the lungs? Well, our pH changes. So we have to have a dependence on H+. This is going to allow for that release of the CO2 and the reabsorption of O2. So depending on the pH, CO2, and I don't remember reading about the chloride actually, and BPG, we're going to end up seeing a different fluctuation between these two. BPG plays an extra interesting role, particularly when you're looking at babies, okay, in the womb. What we're looking at is a difference in concentration of BPG between the fetus and the mother, and that's going to allow for a better transfer of oxygen from the mother's bloodstream into the baby. Why is that going to be important? Sure, we like babies.
Diffuses.
Why do we need that diffusion? Can babies breathe?
No.
No. So the mother is breathing for the baby. That oxygen comes into the bloodstream and has to get transported to the baby. Okay? So the baby has to have a tighter affinity for oxygen so that it can pull the oxygen away from, ultimately, the mother. Okay? So you have a little parasite inside you. Babies. Okay. Lucky you. Okay? So we could take a look at this reaction, okay, where we're looking at the binding of hemoglobin between CO2 and oxygen. So we're ultimately fluctuating back and forth between these situations. If we're in an actively metabolizing situation within the muscle, we need to push that oxygen off the structure and replace it with CO2. Okay? So you can see where those concentration gradients are going to play a role in that overall reaction. Okay? What?
The hemoglobin.
B? Oh, yeah. That's just the symbol they decided to use for hemoglobin. Yeah. Okay. What's that?
You said HG was big.
HG was taken, yeah. Probably is a true story. When it comes to the sapling homework, you will be dealing with some graphs looking at the flow of oxygen across a couple different membranes. Okay, there isn't really anything for me to describe with those beyond look at the graph and interpret what's happening to the concentration of oxygen, your percent of oxygen or saturation, and the amount of oxygen pressure. Okay, and decide where in the body you're located at that time as well. Okay, so if you're in the muscles, we want the saturation of oxygen to be relatively low, so it's going to start shuttling it off somewhere else. Okay? So a lot of the sapling questions, pretty much anything with a graph, I'm going to keep in, regardless of the detail that we're going to talk about it in class, because you should be thinking about the balance between those things. Okay? Does it kind of make sense? Okay. So here's a situation with our fetal hemoglobin. I dropped it right in on top. You'll notice that our fetus, or your fetus, sorry, has a higher affinity for our oxygen. It saturates much more quickly than maternal, there we go, that's the word for it, than our maternal hemoglobin, because again, we want that oxygen to transfer over to the baby or to the fetus instead of staying solely with the mother.
So you're saying that hemoglobin in the fetus is more efficient?
Binds oxygen tighter. So efficiency is going to be a separate issue as far as how we want to decide what we're measuring for efficiency. It needs to be able to pull the oxygen away. Okay? Once the oxygen's away into the baby, it still needs to, or into the fetus, it needs to be able to release that. Okay. Questions? Okay. So for the lab people, what can cause a protein to stop functioning properly? Okay, it's ultimately going to come all the way back to improper folding. So what causes improper folding? pH is going to affect it. Okay. If we go with the broader strokes, if we change the intermolecular forces, period, everything else changes. Okay. How can we change the intermolecular forces? We can put hydrogens onto the structure, take hydrogens off of the structure. Okay, so we can change the pH. What else could we change? Temperature. We increase the temperature, we tend to get reactions to go faster, but the problem with biology or biochemistry is that most of our proteins are focused around a particular temperature range. If we exceed that temperature range, we're now going to cause all of those forces, those intermolecular forces, to now oscillate and break apart. Okay, and we prevent the folding properly. What else could cause protein to come apart? We could look at different solvent effects. Okay, is that going to be as big of an issue when we're looking within a cell? No, because all cells are functioning in water. But if we were going to try and study an individual protein, we're going to take it out of that environment, which means we could potentially be changing the solvent. Okay. Usually not a good idea, so we almost always run with water anyway. Okay. But that's where we'll also bring in our buffer systems to maintain that pH in the appropriate levels. Okay. So again, what we're looking at is the proper folded state, or our native state, versus the denatured state. Okay. In the denatured state, we're looking at a variety of variations of denaturation. It could be taken all the way down to just a primary chain, okay, or the primary structure. Or what we could have done is just disrupted the tertiary structure enough that it can't function properly. Okay. If we continue to denature it, we could potentially destroy the secondary structure. Like I said, take it all the way down to the primary structure, where we have virtually no structure within our amino acid sequence. Okay? So that's what's happening when we denature. Is it possible to get it to renature? In certain circumstances. Okay, so it depends on the proteins. Some proteins are not, or rather robust, so that once we denature them, as long as we remove that denaturant, it can then refold on its own. Okay, so it's looking at ultimately just its interaction with water and the protein itself. But there are other proteins that need chaperones to help put it together in the appropriate space or the appropriate arrangement. If those chaperones aren't present, we're not going to get it to refold. Okay? So a lot of this refolding process can be a bit tricky. So we've got to be careful with that. Remember, what else caused things to fold? It's not just the protein sequence, but amino acid sequence, but that's not going to change with our... Okay, we could look at chaperones, but back to the initial slide that most of you weren't here for. What did our image have in it? So if we go all the way back to our tertiary structure, oh man, it's way back there. The hydrophobic interactions are always going to be there because that's the amino acid sequence. We're not destroying those. Right. The metal. If in the process of isolating our protein, we completely denature it, that metal ion is probably going to float out into the buffer. And if we remove our solution, swap out our solution to remove the denaturant, we're probably also removing the metal ion. So we might also have to put more metal ions back into it to help that folding process. Okay. So be aware of those kind of limitations. Another one that we could look at would be that disulfide bond situation. Okay. That is an oxidation reaction. So if we've reduced our protein in the process of trying to denature it, we're also going to have to allow it to oxidize back. Okay. Our redox reactions are not particularly spontaneous on their own. They usually need something to help them oxidize or reduce. Okay. So we might also have to add another species in there to allow for that bond formation or reformation. Okay. So our protein folding can be a drastically complex and interesting problem to solve. So now we can go all the way back. So what contributes to protein folding? All of those fun things that we were just discussing. The big question that you'll probably see me ask would be something that's illustrated in these two images. What's being illustrated here? It's looking at the side chains. Okay, and our green structure, or our rightmost structure, what are those side chains supposed to be representing? Why hydrophilic? They're all on the outside of the protein. On our leftmost structure, we're looking at our hydrophobics, those amino acids are all centered towards the middle of the protein. Okay? So it's kind of a neat graphical image in that respect because we can start to see what's happening within those changes. Okay? Questions about, I think this is the last slide for chapter 4. Let's make sure I don't have anything hidden underneath it. I don't. No questions? Okay, chapter 5. Ultimately all about lab stuff. This is the sole exciting slide that we're going to talk about in chapter 5. Okay. How might we take advantage of protein structure to do isolation? So we could adjust our pHs. Okay, that pH can potentially change our R group on the outside. If the R group changes, we're going to change the charge of our overall protein. So we can now potentially separate our polypeptide changes based on charge. So we could put a charge across a column, and our proteins are going to travel according to both size and charge. Okay, so we could get an isolation in that respect. If we had just amino acids, how could we get our amino acids to separate?
PKs.
What's that?
PKs.
So indirectly, what happens with the pKa? So it's not so much the pKa we're concerned about. So it comes back to the pH and the same idea with this, the charge on those. So we want to separate based on charge. If we go to the pi, what happens at the pi? They're all neutral. We would not expect them to travel with that electric field. Okay. So what we're evaluating is changing ultimately the pH to change the charge and then go through and change how these things separate. Okay. Do we have to deal with charge? Those of you in lab should be able to answer that question based on this week's lab. No. What are we separating, or what did we use to separate our amino acids in lab this week? TLC plates, which is ultimately polarity, which I think is a horrible term because it's way too vague. I'd shift it to even further. Intermolecular forces. If you couldn't tell, intermolecular forces are kind of an important aspect. Okay? So we can separate them based on the varying intermolecular forces within those side chains. Why would we evaluate our side chains primarily? That's where our primary difference is going to be. If we look at every amino acid, every single amino acid has a carboxylic end and an amine end. Okay? That's not different. That's not going to allow us to separate our amino acids. We have to look for where the difference is, and that's ultimately going to come back to our R group. Okay? So those of you in lab will get to see a lot more of the general techniques as far as separating things. Those of you not in lab probably won't have to worry about it because probably not in this class for the lab technique anyway. Okay? So, chapter 6. I'm not going to lie, it's going to be an interesting chapter to put together. Both chapter 6 and chapter 7. We're going to bring back some more algebra and mathematics, which is everybody's favorite as far as manipulating things. And even further, that algebra and mathematics is going to come in probably everybody's least favorite section of 152, which was... It was such your least favorite section that you probably all forgot that you studied it. You're looking at the kinetics range. So rate equals our rate constant times the concentration of our reactants. Okay. And you probably did experiments in lab where you studied rates. So you looked at how long it took for a color change to occur. A lot of biochemistry, or at least the lab version of it, is based around that same kind of observation. You time how long it takes for a particular observation, and then you correlate that back through to what the enzyme actually did. Okay? So we're going to be focused a lot around kinetics. We're going to look at both the mathematics, and then hopefully we'll try and look at how you would plot this and you'd see it experimentally. So we'll look at a lot of different graphs to go through and interpret this. The chapter 6 homework, I'm pretty sure I made available. I don't think there's a lot of graphs in it, unfortunately, at this point. What you will see a lot of are all of the equations. Okay, so it's going to be looking at those primary equations and how they manipulate and how you can work with them. Chapter 7 then takes it a step further. Chapter 6 is really looking at how an enzyme works appropriately, okay, and it's a standard, easy state to look at. So enzyme plus our substrate gives us a complex, which gives us our product. Okay, very straightforward. Is there something else that we could add to that to make it more difficult? How about something that prevents the enzyme from binding the substrate? Okay, well, how does it prevent the enzyme from binding the substrate? Okay, depending on what mechanism it's working with, you're going to see slightly different results in your kinetics. Okay, so we're going to try and look at those and evaluate how those relationships work, and then talk about the overall mechanisms behind them, and all the fun names that they go through and give them, which oftentimes are counterintuitive. Okay? So our enzymes are ultimately catalysts. They're going to speed a reaction. So if this was our reaction, what would an enzyme do? It's going to lower our activation energy. And this is something that killed me in the book. Okay, to lower the activation energy, are we looking at thermodynamics? No. The energy between our reactants and products stays the same. Okay, the only thing that's different is the energy of our transition state. Okay, that's what's happening with our catalysts. Okay, the next thing that kind of killed me with or this really came with the textbook slides. They went through and did this long discussion of Gibbs free energy and all this other fun stuff. What is Gibbs free energy going to have to do with? Thermodynamics. That's not our kinetics section. Okay. The next thing that I thought was a bit odd is they brought in this idea of temperature, and they correlated it as a catalyst. Is temperature really a catalyst? Not really. It does speed the reaction, but that doesn't necessarily make it a catalyst. What does the catalyst do? It lowers the activation energy. What does temperature do? It increases the ambient energy of our reactants so that we can get over the exact same transition state. Okay? So temperature is not really a catalyst, even though it does speed the reaction. Okay, so kind of be aware of that nuance within this. Questions about that? Because I personally think that's a little bit confusing to reference.
What does that play on?
Huh?
What does that play on?
I don't think I saw any temperature questions in sapling. Okay. I'm not positive on that, but I'm pretty sure most of them went to other things. Okay. So realize what our temperature is doing. It will speed the reaction, but it's not changing the activation barrier, which means it's not officially a catalyst. Okay? Questions about that? Okay. Where do our enzymes come from, actually, before we move forward? We don't make them. Our bodies go through and make them. Ultimately, what we're looking at there, in most cases, our enzymes are proteins, but they can also be RNA, and I think in a few cases, DNA could almost act as an enzyme. Okay. It's a little hand-wavy on that one, I'm pretty sure. But what we're looking at is how some structure can force a reaction to occur. Okay, so pretty much anything with structure could allow for a reaction to occur. Okay. Our primary focus, though, is with our proteins and RNA. So if we look at this qualitatively, there's two main methods or two main theories when you go through and look at enzyme catalysis. You get the lock and key method. So the substrate is effectively your key, and the enzyme is your lock. So as long as the substrate matches the key shape, they fit together, and we can now form our enzyme substrate complex, and we can potentially spit out a product. Okay? So they have to be exactly the same. How can we potentially use, in fact, this works for both of these, so let me step away from that. The other method is our induced fit mechanism, and in that case, I know this is very, whoa, I don't want to go to that yet. I know this is a very subtle change, but what's happening in the induced fit mechanism?
It's like curve one.
You'll notice that your positions 1, 2, and 3 don't match as sharply to our substrate. So what happens is as the substrate comes in, the enzyme changes its shape to match the substrate structure. Okay? Not only did the active site change, but what else changed? The rest of the enzyme changed as well. Okay? So in an induced fit situation, once the enzyme binds something, okay, we won't even say it's necessarily the substrate, the enzyme structure changes. That enzyme structure change could result in producing product more efficiently. It could also result in not ever forming product. Okay? So our induced fit is ultimately a lot to do with the protein changing. Our lock and key is pretty stationary. Any idea which of these you think is probably more prevalent? I would guess the induced fit as well. Okay? Once we put a secondary feature into our structure, more than likely it is going to change its structure. Okay? How significant that change is would be the next issue on deciding between lock and key and induced fit. Okay? Let's say we were trying to target a drug. Okay? In the synthesis of a drug, what are we going to look at?
Depends on the drug, but like antibiotics, it's to either prevent like them growing or prevent like part of their metabolism by binding
So
into protein.
typically we're targeting a protein. Okay? And we want to either shut down or enhance the function of that protein. I want to change the structure. My drug should match the active site. Okay.
Or block it like that.
Or block it. But for it to block it, it still has to be able to fit in that active site. So if I build a drug along these lines, is that going to hit in my enzyme? No, it doesn't match the active site region. It's not going to target, and it's not going to allow for a blocking of that enzyme function.
But if it's non-competitive, it changes confirmation.
That's going to be a slightly different issue, because what we're going to be concerned about there is where our drug targets somewhere else in the enzyme to cause that structure to change. Regardless, where it targets must match the shape of where it's targeting. Okay? So let's say I build a drug that has that exact shape. Okay, well, if it has the exact shape, that's going to be a bit problematic if we're trying to shut down the enzyme. Because it binds, but it has the exact shape as our substrate. All we're doing is increasing the concentration of our substrate in the bloodstream. If anything, we're further activating that product metabolism. Okay? So if we're going to design a drug, we do want it to match that shape, but we don't want it to be exactly the same. So what will we do? We could go through and change the structure on the outside. Give it little devil horns, why not? Those look more like pig ears, but close enough. Okay? So what ends up happening is the substrate comes in and binds, and because of these extra features, that now shuts down or prevents the enzyme from functioning properly. Okay? We don't want it to continue through. So instead of our enzyme now releasing that substrate, it's now become a dead structure, and it can't function anymore. And so we can limit the production of that enzyme. So a lot of drug synthesis is aimed, at least very specific drug synthesis, is aimed around matching your target structure as much as possible, and then doing some slight tweak or manipulation off of it to make sure that it can now no longer be released or no longer be metabolized appropriately, which means that enzyme shuts down. All right. What happens after it binds? Okay, it's going to change the shape of our substrate and release our product. So we end up making our new product and then spitting that piece out. Okay, so this is, again, in our simplest situation. It's a single substrate changing into a single product. Okay, are all reactions just a single substrate? No. In a lot of our cases, we're dealing with two substrates coming together, and our enzyme helps hold those physically close enough to each other to allow for that reaction to occur. Okay. Again, we want to simplify as much as possible. So when we look at the kinetics, we're going to focus on a single substrate and a single enzyme. Okay? And we're really looking at this interaction and how it can propagate forwards. Questions? Okay. So if we take a look at the overall parts of our enzyme, we've got our substrate, we've got our active site. Those two things are going to interact with each other using intermolecular forces. Okay, we will typically not see a covalent bond form. Why no covalent bond? It's not going to detach. Okay? Okay? So that could work in the cases of a drug if we wanted to shut down that enzyme. We make a new covalent bond within that enzyme substrate complex, and now the enzyme can't release the substrate. Okay, so the enzyme becomes dead. All right? So we're looking at relatively weak forces, hydrogen bonding or hydrophobic interactions between those two substrates to allow for that interaction to occur. The stronger the binding, probably the stronger the force. So we might see more electrostatic or more hydrogen bonding in a tighter complex. All right? So the overall ways that we'll abbreviate this, we're looking at E for enzyme, S for substrate, and then we have the ES, which is our enzyme substrate complex. What happens after we form our enzyme substrate complex? Okay. The enzyme substrate complex will dissociate into our enzyme and product. Okay. Let's see, did I actually include that? I did not include that, so let's draw that on there real quickly. Okay. We will see this equation again. Is there anything interesting about this equation?
It's on the reverse form, so.
There's a reversibility associated with this. Once we form the enzyme substrate complex, it doesn't have to form the product. It could very well spit the substrate right back out with no reaction occurring.
That product is available?
There's all sorts of reasons behind what could happen with why that would occur. Okay, but we end up having an equilibrium here. We can both form the enzyme substrate complex, and then we have two ways for the enzyme substrate complex to dissipate. It can either go back to our starting materials, our enzyme and our substrate individually, or it can form our product. All right? So we have to be aware of both of those situations. So if we're going to go through and look at the mathematics and the kinetics of this, what's going to determine the formation of our product? Right, the quantities of each, and then also the rate constants for each of these individual steps. The rate constant for both the formation and then the rate constant for the dissociation back to our enzyme and substrate, or the rate constant for the formation of our products. What's that? I didn't invent it.
Is there reversibility on the complex and the product itself? Or is that a
No, that's a one-way street. Okay, well, we assume that to be a one-way street. Okay, so really we're looking at a balance of three rate constants and how they all interplay with each other to generate an overall reaction. Okay, so we could be concerned about the concentration of our substrate and the concentration of our enzyme. Okay, so those are going to be the two things that we will target the most when we get into the details of our kinetics. Okay, before we go into that, we can take a look at, this comes back to that drug targeting idea. I believe I got these abbreviations right. I just found a cool video, so I wanted to show this. PNP and DADME, we'll stick with that since I don't know the full names. The PNP is a purine nucleoside phosphorylase. And this is, again, I'm not positive because there's a couple PNPs out there. But it's an enzyme that catalyzes the reaction shown here. What we end up doing is taking adenine, and we turn it, or sorry, adenosine, and we turn it into adenine. Okay, so the enzyme catalyzes the cleavage of which bond?
Nitrogen, oxygen, or nitrogen, carbon.
Our nitrogen carbon bond in here. Okay, so our enzyme is going to have to facilitate that breakage. So if we want to shut down this enzyme, what we're going to want to do is build a drug that can somehow mimic that structure, our starting reactant structure, and then hope that that can prevent that interaction. Okay? So here's a bunch of potential drugs that were created with the intent of targeting this enzyme and shutting it down. You'll notice that you're seeing a lot of different structures here. Okay? Only a few of them have structures that even, okay, didn't do anything. Only a few of them have structures that even mimic that adenine structure. The rest of them seem to be apparently not even realistic. Okay? Why?
Because they're synthesized.
The enzyme is probably not grabbing a hold of the adenine or adenine. It's grabbing a hold of some other portion of that structure. And so our drug is targeting where the enzyme grabs. Okay? How could we go through and predict this or see this actually work? Well, we could set up enzyme kinetic studies, and this is probably my least favorite method of biochemistry, but it's very effective. Okay? We can do almost a shotgun approach. You invent a whole bunch of different structures, okay, and then you build a 96 well plate, and you put your enzyme into it, and you put each of these little drugs into it, and then you let it run. Okay? Whichever one seems to shut down that activity the most, you go back and look at that structure, and you start making changes to it, and you set up a new experiment with 96 new structures, and then you again watch which one of those shuts down the protein the most. Okay? And then you continue to refine this until you build or design a structure that seems to have the tightest reactivity or the tightest shutdown of reactivity. Okay? So that would be kind of the experimental method to doing this. It is a very common method. I just don't particularly like it because my analogy is you're using a shotgun and you're shooting at a barn. Okay? You use the shotgun to shoot at the target on the barn. Oh, my bullets went over here, so I need to change my pattern, okay, to try and hit the target a little bit better, as opposed to just figuring out exactly what it is, okay, to hit it. Problem with figuring out exactly what it is, you'd have to know the structure to be able to target it exactly what it was. If we can't determine the structure of the protein, we're kind of stuck with this kind of shotgun method. Okay? So the other option, whoops, I have to clear out of that, is we can model. No. There it goes. Okay, we can model it. So what we see here is, I believe, the DADME drug, and what we've got out here is the protein that they've then modeled on a computer with the overall interactions within the active site. And so what they've gone through and done is started to just model all the possible interactions that can come out of it, just like we saw with the protein folding. Right? And what they're trying to see is how that drug can get intercalated into the active site and how well it binds. Then they can go through and change the structure of the drug, say what happens if it was this shape? How is this now going to bind or interact within that site? Okay? So what they're using is computer modeling to go through and predict this. Okay? And if you believe them, it does a really good job. Okay? In this case, this is a more interesting protein or active site. Our active site is actually buried on the inside of our protein. So the substrate actually has to come and kind of, whoa, hello, dude, weave its way into the structure, okay, through all those kind of interactions. So our proteins are constantly moving. They aren't this single shape. They are constantly moving and flipping. And in those motions, we can sometimes bring our drugs or our target substrates into the system. Okay, and I may have just lost connectivity. Nope. Okay. Another example for our enzyme is you can look at chymotrypsin. It catalyzes the cleavage of the carboxyl side of phenylalanine and tyrosine residues in a peptide. Okay. Any guesses as to the characteristics of the active site in the enzyme? What do phenylalanine and tyrosine have in common? They both have that aromatic ring within their structure. That aromatic ring happens to be characteristic within the active site of this enzyme. So if you go through and look at exactly where it binds, there's a bunch of hydrophobic interactions in the enzyme that line up with that phenylalanine and tyrosine side chain. So it uses those to kind of grab a hold of the protein, and then it can cleave the bond. Okay? That cleavage, it's not just grabbing those, but it has to grab them with one hand and effectively cut with the other, which means it's going to be very specific to where it cuts. Notice it's cutting only the carboxyl end. It's not cutting the amine end of those chains. Okay? And it's again because of where that phenyl group, let's see, did I have a nice picture of this? Okay, it's where that phenyl group sits in our protein. So if we go through and look at this amino acid sequence, where would we predict the cut to occur? it cleaves the carboxyl end of our amino acid. So there's our amino acid for that one's phenylalanine. It cleaves there, and it also cleaves, oh, I didn't do a very good job on this one. Cleaves out there. I guess we should draw the rest of that structure. And it won't cleave where I drew that other parentheses. It only cleaves on the carboxyl end of that particular amino acid. And it will target those two amino acids because those two put an aromatic ring in relatively the exact same position. Whereas if we shifted to, say, tryptophan, tryptophan also has an aromatic ring, but that's now pushed a little bit further out because it also has that 5-membered nitrogen ring as well. Okay. It's kind of an interesting enzyme. I was trying to find some references to it, and I couldn't quite track them down. I know Sapling has some questions around this enzyme that I've seen in organic classes, but I didn't see any in your homework. It's used to determine peptide sequences. So it degrades proteins, so it's, I can't think of the name. But what it'll do is cut up proteins. So if we're trying to figure out a protein sequence, if it's too long for us to interpret, we can go through and cut it down. So we can use these degradation enzymes like chymotrypsin and trypsin and a couple other ones that will cleave our protein into smaller sections. We can then evaluate the sequence of those smaller sections a little bit easier, and then we can put those sections back together once we figure out what everything is. Okay? And it has to do with exactly where they're cleaving. So it's kind of a neat enzyme in that respect. Goody, kinetics. So, given the reaction, what is the rate of the reaction with respect to, so that's WRT, A or D? Let's see how you guys remember this. Whoops, so rate equals. So we could go through and look at concentration and our rate constants. Okay, but is that going to work for D? It's all about our reactants, right? So if D is a product, how can we get a rate?
We do the forward and reactant.
Our forward and reverse. So what we're looking at is the rate is going to be the concentration of A, or ultimately the change in the concentration of A with respect to the change in time. Okay. We could also go through and say our rate is going to be related to the change in the concentration of D with respect to time. If I want to set these two equal to each other, what do I have to do? No. One has to become negative. Which one becomes negative? A is going to be negative. If we're running our reaction, what's going to happen to the concentration of A? It's going to decrease. So our rate would be negative. What's going to happen to the concentration of D? It's going to increase. So we get a negative equals a positive. That doesn't work. So we have to bring a negative into it. Our standard is to make our rates positive just because we hate negatives. Okay, so that negative usually shows up in front of our concentration of A. What if we wanted to figure out a rate constant? Okay, so now we could bring in a rate expression. And I think I must have done too much copy-paste here because that's not asking what I wanted it to ask. Now we can say our rate is equal to our rate constant times the concentration of our reactants to some power. Those of you in my class, I ended up using M times the concentration of B to the nth power. Okay. Officially, it's not just reactants, it's all species. Okay. Why did I use M, N, O, and P? What are those? True, they are letters. Those are the orders
Oh, yeah.
with respect to those reagents to the reaction. Okay. M and N are typically some whole number integer. What are O and P typically? Technically a whole number integer, but it's a very specific one.
0.
0. It's usually 0 because our products typically don't have an effect on the overall reaction. Is that true, particularly in biochemistry? No. Oftentimes our products will feed back into the reaction and shut it down or speed it up. Okay? So when we look at biochemistry, our rate expressions can get really complicated because the orders with respect to our products don't always equal 0. Okay? They can feed back in. So what we're looking at is our overall rate expression, and then we want to be able to interpret our rate because what we want to know is how can we make the reaction go faster or slower, or in the case of biochemistry, how does the body or cells make that reaction go faster or slower? Okay, so we want to understand those limitations within it. Okay? So if we went through and took a look at a plot, we could plot the rate versus the concentration of A. I just had to drop that so you could see it. Okay, and we could end up seeing some different versions of this. What do those versions mean? Okay, so we see a red line, a blue line, and a green line.
Blue line means concentration doesn't matter, a red line means that it increases with
If the concentration doesn't matter, what does that mean? What is the order with respect to A?
1. Oh, 0.
For blue, it would have to mean the order is 0. There is no relationship. And it's not that it's linear, it's that it's flat.
Yeah,
I talked about the red
Yeah, the red.
If we move to the red one, what happens in the case of the red one?
1.
Our order becomes 1 because there's a direct relationship. As we 1-for-1 increase our reactant, we get a linear relationship with our rate. It speeds up. What happens with the green one?
That's the second order.
Okay. In the case of our green one, it's not a linear relationship. You notice it's going increasing exponentially. To generate that curve, we're looking at a second order. Okay. You guys, in theory, have a little bit more mathematics and plotting ability than we did back in the day, which means we couldn't generate these kind of nice curves. And our data probably looks something, particularly for that green one, more along the lines of things kind of dancing all over the place. Okay, maybe being close, maybe being way off, maybe being right back on the line. Okay. So it became very difficult to interpret that curved shape to be a second order reaction. So what do we end up doing? Nope, line of best fit only works if you got a computer to be able to calculate that. No.
Don't they one of them counts as the other, like the outliers?
What we want to be able to do is to convert the data into a format that we can see very quickly and easily. That green line has a slight curve to it, but that's because I dropped a line on it. The blue line, does that have a curve to it?
No.
Absolutely not. You can see that. Why can you see that?
Because they're all you get the
It's
same 40.
straight. A straight line is easier for our eye to naturally see and determine, particularly if we've got all sorts of data points bouncing all over the place. Okay? Which of course we're going to have. There's error in our reports, okay, or in our experiments. Okay? So what we try and do is reorganize our equation. Our rate equals k to the a, or k times a to its order times b to its order. We want to reorganize that equation into the equation of a straight line. Once it's in the format of a straight line, we can now interpret it much more quickly and cleanly. We can calculate the slope, we can calculate the y-intercept without having to deal with these exponential features. Okay? So we're always driving towards straight line dynamics. So when we get into the Michaelis-Menten equation and we look at that, you're going to find out that it doesn't look like a straight line. So don't blame me. Somebody else had to come through and say, okay, how do we make that look like a straight line? Okay? And that's where you get your Lineweaver-Burke plots. So what we're doing is interpreting relative the simple aspects of our 152 and now making it drastically more complicated, almost drastically more complicated. Summary of our orders, okay, just in case you forgot. If we're looking at a reagent order of 0, it's not having an impact, so I would usually call it a bystander or a spectator in the overall reaction. Order of 1, it's involved in the reaction. Order of 2, it's really, really involved in the reaction. Order 3 it's typically only that species. Why is it only that species?
Everything else is.
Can we get fourth order?
No.
What does it mean for each of these numbers? 0 means not involved at all in the slow step. What does 1 mean? It's involved in the slow step. 2 means? What does that 2 directly mean beyond super involved? It means 2 molecules are involved in that slow step. What does 3 mean? 3 molecules are involved in the slow step. Why do we not see 4?
4 is a crowd.
Close enough. Said probably a little bit nicer than I could. The likelihood that we can get 4 different molecules all coming together at the exact same instant to allow for that bond transformation is very, very unlikely. Okay. Our first order and our second order are much more likely to occur. Okay. Even the third order starts to push the boundaries of what we can get to happen. Okay. You can think about couple or populations of people in the world. We got a lot of single people, quite a few couples, and then usually we don't get very many people holding together as groups of 3 for very long. Okay. And then it does happen. And beyond that, it really gets a large stretch, a very small percentage of the population, probably so small that we could say it's 0%, gets 4 or more people all coming together and being able to live with each other reasonably. Okay. So, what can we do with our enzyme kinetics? Okay. So if we look at just the substrate and products, so I'm actually kind of being nice and easing into this a little bit slower than I thought. If we look at just forming the substrate from our product, okay, what we're going to end up with is what's supposed to be a nice straight line there. Okay. As we increase the concentration, our rate speeds. Okay. And what we're assuming in that situation is that our rate is going to be equal to our rate constant times our concentration of A. So we're assuming first order dynamics in this. What happens if we throw in the enzyme? It speeds it up. Okay. How is it going to speed it up though? So it's going to change the energy of activation, but how does that change our curve? We would expect it to be a sharper line. Should it be exactly linear? No. Why is it not going to be linear? The enzyme will speed the reaction so long as there's enzyme available to react with substrate. What happens if we increase the substrate concentration so much that we've run out of enzyme to bind? It's going to plateau. So what we would expect to see with our enzyme and our substrate is probably a very sharp line initially, but then eventually it's going to flatten off. Okay. Yes?
You're not talking like the enzymes used up.
Not that it's sort of. It's already in the process of being used. Okay? So remember, our overall reaction was our enzyme plus our substrate goes to our enzyme substrate, which then goes to our enzyme plus our product. And really what we're evaluating for our rate is our product.
The rate always goes down to like as soon as it releases it, it can only grab one.
It can only take on one more. So at very low substrate concentrations, there's enough enzyme that it immediately reacts with our substrate to form the enzyme substrate, and it can form our product right away. But what happens if we increase the concentration of our substrate beyond the concentration of our enzyme or the capacity of our enzyme? Now the substrate is just going to hang out and wait until there's an enzyme available. So our rate's going to flatten out. Okay? We'll talk about that later too.
So
Yeah.
it's more important to not really have the amount of substrate, but more the concentration of the enzyme.
It's a balance between them.
Okay.
It is a balance between them. And so what we're ultimately trying to figure out is some information about the enzyme. So when we're looking at a study, we're going to put in an X number of enzyme, and then we're going to change the substrate concentration. Because if we're going to take a look at this curve, where is biology going to typically run its reaction? Where's its balance between substrate and enzyme?
Right before the inflection?
Probably going to go right before it curves off. Because after that, I have a whole bunch of a whole bunch of substrate floating around that could potentially do hazardous things. So our biology is probably going to start to run the reaction maybe in this system, where we get a maximum rate return, and we don't have a buildup of one species over the other. Okay? So what we're trying to do is go through and see where that return is. To be able to do that, we have to generate these kind of plots and be able to interpret them. Okay? What happens if we have cooperative binding, say in our hemoglobin?
It's deeper, but like it flattens onto it.
Cooperative binding means we bind one, and then the next one binds even faster. Okay, so I'm going to have to accent this a little bit weird, but it's going to happen anyway. It's going to go sigmoidal. It's going to start relatively low, but then as we put stuff on, it now all of a sudden is going to bind much more quickly to our substrate, and then again it will level off. So we'll end up with some sigmoidal type shape. All right. So based on a plot of our rate versus our concentration, we can start to predict some information about how our reaction's occurring. We won't ever really look at the R1, or sorry, R. I don't know why I said that. Our red one. There we go. That's why I said R. Right? We aren't going to look at the red one because in biochemistry we're running these reactions with an enzyme. So what we're primarily concerned about is, does our enzyme function in just kind of a standard form, or is it a non-standard form? Are we seeing some kind of cooperative binding? So a simple experiment of plotting our rate versus our concentration could allow us to interpret some information about how that enzyme functions. Okay, kind of makes sense? Next issue with this. Okay, if we're going to try and determine a rate expression, okay, as we've got down here, does that equation work for either the blue or the purple lines? Okay, why not? Okay, that's a linear expression. Okay, if we look at our blue line, we could probably break that down or approximate it into two situations. At first, what do we have? A first order relationship. Then what do we have? It shifts to zero order. I don't know. Zeroth? There we go. Zeroth order. And our sigmoidal one, that's going to be even more complex as far as how that approximates out. We can look at one section of it where, yeah, we're looking at first order. But then what's happening with the rest of this, we've got a zero order early and a zero order late. Okay? So we're going to get a very odd kind of composition of how we would predict those. Okay? So let's actually, why is my screen not catching up? Oh, probably because my screen is not connected. Okay, there we go. So remember, our enzyme reacts with our substrate to form the enzyme substrate complex, and then our enzyme substrate complex can break down, right, into either the substrate and enzyme or the enzyme and product. So what we're looking at for our overall reaction is what's shown in red there. Okay. How can we go through and determine our overall rate? Okay, because that's our target. We want to know our overall rate. How do we do it in 152?
We had to change one and keep one constant.
Yeah. Okay. If we assumed that our second step was the slowest, we said we achieve a pre-equilibrium and we had to solve for a concentration of the enzyme substrate complex. Okay. Ultimately, in this case, that's what we're really going to be concerned about. We're going to assume, or we're going to say that the formation of our product is really going to be dependent on that last section. Okay. But do we know that K2 is substantially slower than K1 or K minus 1? No. So that's what makes this a lot more difficult to solve. We can't just go through and do direct substitutions from that rate expression. Okay, ultimately, what we're looking at is rate constants that could very well be very close to each other. We can't go through that pre-equilibrium substitution. Okay, so what we're going to go through and do is think about our rate. It is dependent on the concentration of S, okay, as it converts over into our enzyme substrate complex. So at low concentrations of S, our rate is going to be equal to our K2 times our enzyme substrate. Okay, why? Because at low concentrations of S, the enzyme immediately binds it. We have a relatively fast K1 to immediately form our enzyme substrate complex, which then can dissociate with our order of K2 to generate our product. Okay, so at very low concentrations, we end up with that relationship. At very high concentrations of S, we're now going to be dependent on the enzyme that's present. In fact, the total enzyme concentration. Because remember, what happens at high S, we've put in so much extra substrate that it exceeds the capacity of the enzyme. So our rate is now going to be dependent on our enzyme concentration and less on the enzyme substrate concentration. Okay? So now that we've got these two dynamics, we want to try and play around with them and see what we can come up with. Okay? Big issues that we've got. Do we know the concentration of the enzyme and substrate complex? No. When we set up the experiment, we can put in enzyme and we can put in substrate, and that's about all we can put in. Okay? We could potentially control or see how much product comes out, but we have no idea how much of that substrate actually formed the enzyme substrate complex. Okay? So initially, that's going to be a problem. Okay? We could also look at our high substrate concentration. We'd have it dependent on our enzyme total, how much enzyme we started with. Well, that we would know, but then do we know K2? No, because our K2 is going to be dependent on our enzyme substrate. Again, we've got a problem. Okay? So this is where Michaelis and Minton came through and started to manipulate these things. Okay? So what we're going to do is assume that we reach a steady state. What rates are going to be equal to each other?
K1, back and forth. And reverse back and forth.
Not quite. That assumes they reach equilibrium. But they're reaching equilibrium with K2 at the same time. Okay? So what we're going to end up assuming in this case is that the rate of the formation of the enzyme substrate complex will be equal to the rate of the decomposition of the enzyme substrate complex. That's when we achieve steady state. So we are assuming that we're at steady state, and we have to make that assumption. Otherwise, everything falls apart. And as far as whether it's a big leap or not, how would we figure that out? Test it. See if it matches. If it matches and it works, then hey, it's not that big of a leap. And it turns out it actually works out pretty well. So what is the rate of the formation of the enzyme substrate complex? It's dependent on... Depends on the concentration of enzyme and substrate and K1. Okay. We're looking at the formation. If we look at our reactions, the only one that forms our enzyme substrate complex is our K1. So our rate for the formation of the enzyme substrate complex is going to be K1 times the concentration of enzyme times the concentration of substrate. What is the rate of decomposition of the enzyme substrate complex?
It should be ES.
So concentration of ES times K minus 1, true, because that's looking at the reverse. But is that the only thing that looks at the decomposition?
No,
K2 is also involved in the decomposition of the enzyme substrate. And this is where it differs from that pre-equilibrium that you guys were suggesting. Okay? Instead of it just being one simple system, we get an expression with two rate expressions involved in the addition there. Okay? Which makes this a little bit trickier to manipulate. Okay? With this, can we do any simplifications?
Your ES. I don't know. Your K1, K minus 1.
K1 and K minus 1 don't necessarily cancel out. Remember, we don't know what those numbers are. Okay? And they aren't necessarily equal. So we could simplify that out into K2. There's my pen. And then I know it's all exciting, but I think we're probably going to have to stop pretty quickly here.
No.
Times the concentration of our enzyme substrate complex. Okay, yeah, we could simplify that. What is K2?
K2 is in decomposition.
Give me a name for K2. It's the rate constant for the decomposition of the enzyme substrate into the product. It was the rate what? Constant. What is enzyme 1 or K1? Minus 1. It's a constant. What's a constant plus a constant? A constant. We can now go through and collect our constant terms. Just like if you think back to the transition between the kinetics and the equilibrium chapter, we looked at the rate of the forward and the rate of the reverse. and we set them equal to each other, and we collected all the concentrations on one side, and we collected all the rate constants on the other side, and what we solved for was the equilibrium constant. All right? The relationship that we get out of this is this new expression. Because I have a constant plus a constant divided by a constant, that is really equal to a constant. It's not 1 necessarily, it's going to be a constant. So this is where we introduce a new term. That new term is Km, or the Michaelis-Menten constant. Okay? And then I'll go ahead and stop it with that. Okay? Feel free to take a look at the textbook to try and see the rest of that propagation. What we're going to end up deriving is the Michaelis-Menten expression and also the Lineweaver-Burke expression. Okay? So we'll finish that derivation next week.